Hello, my name is Xavier Thayanararia from Bioprocess Engineering, and I will be talking about DNA replication and protein synthesis. Okay, first, what is DNA replication? DNA replication is the process by which a cell creates an identical copy of its DNA before cell division. It ensures that each daughter cell receives an exact copy of the genetic material. Occurs during the S phase of the cell cycle. This process is essential for growth, repair, and reproduction in all living organisms. The steps of DNA replication is first the initiation, where helicase unwinds the double helix at specific sites called origins of replication, creating a replication fork. Single strand binding proteins stabilize the unwound strands to prevent re-annealing. Next is the elongation, where primase synthesizes short RNA primers to provide a starting point for DNA polymerase. DNA polymerase adds complementary nucleotides to the template strand. The leading strand is synthesized continuously, while the lagging strand is synthesized in fragments. Lastly is termination, where sonuclease removes RNA primers and DNA polymerase fills in the gaps with DNA nucleotides. Ligase seals the gaps between Okazaki fragments, forming a continuous strand. Next is key enzymes in DNA replication. First is the helicase, which unwinds the DNA strands, the primase, which synthesizes RNA primers. Next is the DNA polymerase, which adds nucleotides and proofreads. Next is the exonuclease, which removes RNA primers, and the ligase, which joins Okazaki fragments. Next is protein synthesis. Protein synthesis converts genetic instructions into functional proteins through two stages of which are transcription and translation. These proteins perform critical cellular functions like enzymatic reactions, signaling, and structural support. Next is transcriptions, where first it occurs in the nucleus, where DNA serves as a temple for RNA synthesis. Two, RNA polymerase binds to a specific region on DNA called the promoter and unwinds the double helix. Number three, RNA polymerase reads one strand of DNA and synthesizes a complementary mRNA strand using base pairing rules. Fourth, once RNA synthesis is complete, it detaches from DNA and exits the nucleus through nuclear pores to reach ribosomes in the cytoplasm. Translation, which takes place in the cytoplasm at ribosomes. It acts as a template containing codons, three nucleotide sequences that specify amino acids. The ribosome links amino acids together in a specific sequence to form a polypeptide chain, which folds into a functional protein. And these are the key enzymes in protein synthesis, which are mRNA, tRNA, ribosomes, and codons and anticodons.